Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Every so often, a tool emerges that doesn't solve a technical problem. It removes a barrier that the field quietly accepted as just the way it is. Today's episode is about one of those tools. I'm joined by Dr. Eric Trinquet, head of Life Sciences R&D at Revvity. He has spent decades shaping how we study GPCRs, from co-developing the IP1 assay to building platforms like Tag-lite. And now, with the launch of pHSense, he's helping us reimagine how receptor internalization is measured. You'll hear the inside story of how pHSense was developed, from rare earth complex design to the aha moment when internalization could be tracked at endogenous levels, no imaging required. And you'll understand why this isn't just a technical achievement, it's a turning point. You can explore the full pHSense toolkit plus Revvity's GPCR portfolio by visiting the link posted in the description of this podcast episode. And this week in Terry's Corner, the lesson that we launch is a perfect companion. Dr. Kenakin breaks down opposing processes in GPCR pharmacology, including internalization, surface signaling, and why your in vitro readout doesn't always predict what happens in vivo. Dr. GPCR premium members get over 50% off access to Terry's Corner. And I want to take a moment to thank our partners, Revvity and Celtaris Research, and our incredible community pushing the boundaries of receptor science. Everything we discussed today is linked for you below. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. Today, I'm very excited. This is a very special Dr. GPCR podcast episode. And I have with me none other than Dr. Eric Trinquet. Eric, welcome to the podcast.
Yeah, thank you, Yamina. Welcome also you on board.
I'm very excited to talk to you today. This is a very special episode, but before we dive into why this episode is so special, could you please introduce yourself, tell us who you are and what do you do?
Yes, I will. So I'm Eric Trinquet. I'm leading the Life Science Research and Development for all the reagents at Revvity. I did all my career in R&D and especially around GPCR. They are very close to my heart since more than 2 decades. Before joining Revvity, I was part of a biotech called SysBio BioAssay, which has been acquired by the group in 2019. And still at SysBio, I was leading a lot of projects related to GPCR. Among the most famous, I will cite the IP1 assay, as well as the design and the development of the Tag-lite platform, which has been in the 2 past decades, 2 main, I would say, breakthrough in the kind of functional assay you can carry it out around GPCR. And I'm more than happy to be with you today to speak about the latest innovation we are releasing around the functional assay for GPCR.
It's a very exciting launch, but before we talk about it, I'd be curious to hear about your story as a scientist. Were you mentioned that you love GPCRs. We do have that in common. Otherwise, this podcast would not be called the Dr. GPCR podcast. Did you always know you wanted to be a scientist?
Not really. I would say during my graduate school and high school, was not that sure. Business to some extent was in the back of my mind, but for many reasons, I ended up at SysBio having a scientist position in the very early days of the HTRF technology, which is still one of the technology platform we are developing. And at that time, I realized that innovation is something in which I can find a lot in terms of involvement, excitation, and starting first with more photophysical aspect and the development of fluorescent probe. And later on, when we really established the platform, GPCR were and are still one of the most important target class in the drug discovery. So I've been interested in and discovered that behind that family of GPCR, there is so many to do to understand how they are modulated by compounds that it drove a large part of my career as a scientist, I should say.
That is wonderful. And you mentioned, the GPCR field and how the HTRF technology has helped elucidate some of the mechanisms underlying GPCR signaling. And you also mentioned that you were at the very beginning, you joined Cisbio when the story of the company and the technology had started. Tell us a little bit more about the story of the HTRF technology. And after that, we can talk about the new product that Revvity is launching.
Initially, in fact, at Cisbio, we were mainly a diagnostic company. And the first, I would say, development around the technology was really based about establishing biomarker immunoassay to follow up in mainly blood sample some key biomarker. But pretty quickly, we also realized, because of the key feature of the platform, meaning being no wash and can be highly miniaturized, discussing with people at that time from not Sanofi, but the premises of Sanofi, we discovered that maybe the technology can be very efficient to screen and characterize compounds. And it was the golden age of the super high throughput screening platform. And we quickly did some trial applying first the technology to biochemical assay. It was very successful. And immediately after, then we are back to the GPCR. The need of having assay for GPCR, for which biochemical assay is not at all something efficient, raised the question, can we develop functional cell-based assay for GPCR? And then the story around what we did around cyclic AMP IP1 and many other products started.
That's so fascinating. Can I ask, from a timeline perspective, you mentioned you worked on developing the IP1 kit, but was it IP1 or cyclic AMP that came out first?
No, the first was the cyclic AMP. Cyclic AMP was, I should say, easier to develop first because there was already existing, I would say, assay design we have been inspired from. So that was much easier to figure out to develop a cyclic AMP assay. And through the success of it, we said, okay, we need something for GQ coupled GPCR. Calcium was clearly out of the scope for us because we were chasing after assay at the equilibrium. Yeah. So quickly, we have been interested in inositol phosphate and inositol phosphate pathway and regulation. And after, I would say, a long time discussing what should be the design of the assay we should have, we ended up with the idea we can accumulate IP1, which is a metabolite of the inositol 3-phosphate in cells. So let's try to detect it. And then the project itself, I would say, really started at a bench level. Before that, it was more a lot of discussion and research about what we should do. And still, it was one of the largest and I should say risky projects we carried out in the GPCR field.
Yeah, and you have a great point regarding the GQ pathway, because the calcium, especially, I'm assuming that the discussion around this kit was around that period where ultra large screens were something that people were doing, and calcium, as we know, it's not an endpoint assay. You have all sorts of moving parts, and therefore having a GQ IP1 kit was the way to go. I have to tell you this: when I was the master's student, we tried to measure IP3 using radioactivity, and I had the columns, actually 2 sets of columns that I had to pack myself. Yep. And it was just such a messy thing. So having a kit like the IP1 kit is just so much easier. And also it's an endpoint, there's less issues, and you can do it in a plate and not have, I had, I think, 24 columns that I had to put one on top of the other, and the radioactivity was not helping either.
I can fully understand what you said. We did the same because for developing the HTRF version, we need a gold standard. So we also spend a lot of time with these fancy columns and super low and cumbersome, low throughput and cumbersome experiments. Yeah. And but since the first days of that project, we were convinced that we have to go into that direction, trying to stay away to the calcium stuff, which were not at the equilibrium. And it took a while to really implement the assay and convince people that it's really something efficient if you want to deeply characterize compounds and predict more accurately the in vivo behavior of compounds using equilibrium assay. in that context, later on, we collaborated a lot with Terry Kenakin on that, and it was really instrumental to share our experience and how people can use the assay to really do the best pharmacology they can when they were characterizing compounds.
Yeah, and Terry is wonderful. As everyone listening to the podcast knows how amazing he is. Wonderful. It sounds like whenever you guys worked on developing a product, you put in a lot of thought. You looked at what people are doing, what's out there, what's the throughput. And once you had a plan, then you went to the bench and developed that new product and obviously tested it against the traditional standard assay available at the time. And today we're here to talk about you, but also to talk about pHSense. This is a new product that's being launched by Revvity, and we are partnering with Revvity in order to help amplify the voice and help the community use this new product. So tell us a little bit about the story of pHSense. What is it? How did it come to be?
Okay. Two aspects, again, in that project. First one is, since, I would say, decades, we are always working on rare earth complexes, either terbium or europium complexes, having in the back of your mind the idea that maybe we can, at the same time, improve the HTRF technology and maybe expand the application of that kind of very specific probe in some area of the biology. And the pHSense project is a good example of it. We started by designing, I would say, conventional rare earth complexes, meaning they were not at all pH sensitive. And we identify very interesting chemical scaffold. And this is something we did in collaboration with the Professor David Parker from the Durham University, which is one of the best experts in the field. And working around that class of probes, we were figuring out if they can really change, I would say, some feature of the HTRF assay, which was not that obvious, but they were super bright. And we said, okay, with this, maybe we can change a bit the strategy around the development of the chemical entities. And let's see if we can tune their properties by different ways. And very quickly, we realized that having that kind of chemical scaffold, it was pretty easy to tune the molecule into a pH-sensitive probe with two main features. First one is using the same backbone, just by tuning few chemical moiety in it, you can create a wide range of molecule in terms of pKa versus their pH. So meaning you can change their responsiveness. Either they can become more responsive or less responsive toward the range of pH, which was of interest. But the other feature, which was really key, I think, in what we did later on, was the fact that we discovered that molecule, you can create a two-dimensional pH responsiveness. You can play with the brightness of the compound, meaning that you are not changing the spectrum of the molecule, but just its ability to emit light and more light at lower pH. But at the same time, we have been able to tune the lifetime of the product, meaning that at physiological pH, the fluorescence lifetime is relatively short for a europium complex. You are in the range of 100 to 200 microseconds. That's super long compared to conventional fluorophores, but pretty short for rare earth complexes. But the beauty was, if you decrease the pH, you are dramatically increasing the lifetime up to close to a millisecond range. So having that two-dimensional capability of modulating the signal versus the pH, we quickly ended up with the idea that maybe that's really a way to dramatically improve the sensitivity of that kind of probe compared to the conventional one. Wow. Why? Because with the conventional probes, we did investigation at the early stages of the product. And what we have seen is that if you are talking about the red pH-sensitive probe, the orange one, which are the most popular these days, they are okay if you are doing flow cytometry, imaging, not that bad. But as soon as you were moving to a plate reader-based format, the background was impairing a lot the signal to background, and the responsiveness of the molecule was there, but overall, I would say we were not really able to achieve a decent sensitivity. So expectation with the probe we have designed and which are now named pHSense was really to change it and be able to move to the plate reader-based format with something with a sensitivity which would be either equal or better than the one you can get on an imager or on a flow cytometer with conventional probes. And this is what we did at the end by selecting the best probe among the family and implement them into a real assay.
That is so fascinating. And, a couple of more less scientific, but more background story questions I have for you. What people sometimes don't think about is whenever a new product comes out, it just appears on the shelf. You can order it, you have a catalog number. But I want to highlight how much work and how much scientific work and rigor goes into getting to a product. And also the fact that you collaborated, I'm assuming with a professor, I can't remember his name, in academia, was an expert at that. So just want to highlight what you just said and tell the audience that getting to a product means to do a lot of science in the background, making sure that a product holds it works, but also talking to experts. We mentioned Terry Kenakin for the IP1 and working with academic scientists who are experts in their field. And I think that's an important point to make, because oftentimes when you're the end user, you just say, oh, I'm just going to order this and get it and then use it.
No, you're right. This is the way I think for almost every project, really innovative product we are launching, we are following the same path. Paths are basic, I would say, work with the better expert in the world. So on one side, we were collaborating with David Parker for the probes themselves. And on the other side, figuring out how to apply them to GPCR. We have the long-standing collaboration we have established with the Genomic Functional Institute in Montpellier and the team of Jean-Philippe Pin. And for us, having the two experts with us in collaborating and figuring out how we have to select the best molecule for a given application was really instrumental in being able to design something. Because in that kind of system, it's always very good to quickly be able to challenge your ideas and the theory which are supporting your ideas to the real life. And with GPCR and their internalization, which is what we aim to track with the pHSense, we all know for a while that it's something not that well known. There is a lot of aspect. Is it agonist induced or is it constitutive activity? There is a lot of parameter to consider. The model you will select to test this are not that easy time to handle. So having the best expert, being able to create a set of probes, and that was the chemistry part in which David Parker was really instrumental. And when we had available a bunch of molecules, then we said, okay, let's try to apply them to GPCR and let's see what are the outcome depending the brightness, the PK, the lifetime. Every parameter was taken into account, and we assessed them on several models to figure out what we can achieve. And then later on, the main challenge for us, especially with GPCR, was to say, can we get rid of overexpressed system? And that was the later stage, but still we always had that stage in mind, and say, okay, let's go step by step. But what we aim to achieve is to show that we can track the GPCR internalization at an endogenous level. And this is what we fully did with the GLP-1 receptor in some beta cells from rat very recently. So I would say it's a journey, but you need to work with the best expert. You are maximizing your chances of success. And the it's a real partnership, meaning that this is not just saying we will sponsor a PhD or whatever. It's a joint project. And that's also key. There is a personal relationship, involvement. David is still with us for other things, as well as Jean-Philippe, because there is always more to do. And what are the next challenge? That's also what drives me in science these days. So yeah, a nice experience overall.
I think it's wonderful because one, we don't talk about this enough in general about these partnerships. We talk in about collaborations in academia or industry, but having a solutions provider like Revvity and highlighting the collaboration between Revvity and the different academic collaborators, the experts of it all, I think it adds on to appreciating that product, that nicely packaged product that you get once you place the order, because you know that not only internally a lot of work has gone into it, but it's a true partnership and it's a true effort in order to improve the understanding of GPCR biology in this case.
Definitely.
It's wonderful.
It's the packaging, and I would say the commercialization is just the last step of a long journey. Yeah. But the journey has started years ago, and based on ideas. Not all of them were right, let's be honest. But I think we did well. We picked up the right molecule. We developed them properly. Then discussing with GPCR experts, trying to figure out what are the most important, I would say, reagents related to pHSense we have to deal with first. Of course, we are now figuring out what are the next reagents we will release for which purpose and what we can do in a better way. That's for me the commercialization. It's a second step and the beginning of another journey.
Yeah, it does sound. I can tell that you really love this whole journey of discovery and understanding what are the tools that we have now, what's missing, and how can we fill that gap. And I think product development in that sense is just a magnificent journey to get to that, catalog number and that beautifully packaged product there.
Yeah, this is what drives me. I would say for me, the commercialization is an event itself, because it's the way we are celebrating the success of our R&D project. But at the same time, it's the beginning of another journey. We will get feedback from customer. I'm sure that nothing would be 100% perfect. People will ask for more, or I don't know. And then collecting all that feedback is also the way to say, okay, so with what we have in our hands right now behind the scene at the R&D level, what can be improved, what we can do more, what are the new questions the data people will generate with pHSense will raise. And I'm sure there will be. So this is where I think you are trying to figure out what are the next reagents to do. Maybe it can be something very different, or just an evolution of the technology and the probe behind pHSense. We'll see in the future.
I know I can't ask about what else you're working. I mean, I can ask, but you cannot answer. But I'm excited to see what's next. I know this is a podcast and we don't have a visual aid, and I know we're also recording audio and video. But at a high level, if you can walk us through how pHSense works and what are some of the questions that people can answer using this new product.
So for GPCR application, I think the general scheme of the internalization assay you can carry out with pHSense is very easy to understand. You need to attach the pHSense probe to your GPCR of interest. It can be done by various ways. Either you can use a snap tag at the extracellular part, then we are providing substrate of the snap which are labeled with the pHSense. So this is a way in which you can have a covalent attachment of the pHSense probe to the GPCR. If you don't want to use it, we have now anti-flag antibodies, which are labeled with pHSense. So meaning that a lot of overexpressed GPCR in stable cell line are bearing a flag tag. Soon we will release other tag to cover also what people are doing. Can be cyzistidine, it can be HA tag. I think before a year we will release new stuff like this. And the last part is if you want to assess the internalization of an endogenous GPCR, then people need to have a neutral antibody against their GPCR of interest, meaning something which is able to bind specifically the GPCR, but not having any pharmacological effect on it. And we are commercializing a Fab fragment against human IgG or mouse IgG. We are expanding it. So then you can indirectly label this antibody with the Fab fragment. And then in any of these situations, first step is attach the pHSense probe to the GPCR and then stimulate the GPCR by your favorite agonist. And then you can track over time the internalization. You can do either kinetic or endpoint assay, and you will see nicely the internalization of the GPCR.
I think it's beautiful.
Yeah.
I think it's beautiful because it is high throughput. It's a no wash.
Yeah.
And there's no, assuming you have, for example, your stable cell line with your flag tag receptor, the sky's the limit. Especially if you have the good robots to add the reagents and the compounds, you can run as many plates as you can in an 8-hour period in a day. So that's a lot of compounds. And pairing it with other tools, for example, HTRF products, I think it gives you a nice coverage of the different pathways that GPCRs can activate.
That's definitely, I would say, for drug discovery, we all know these days that there is super interesting bias ligand between their G protein signaling and their bias towards arrestin and internalization, arrestin dependent or independent, whatever is the situation. So we are providing that kind of tools, but also at the academic level, I think playing a bit with the technology and the temperature also, you can get a lot of info about constitutive activity versus agonist-induced activity. What are the drivers of it? With the team of Jean-Philippe Pain in Montpellier, we are started to have interesting stories around it and more focus about the understanding of how GPCR are really functioning at the cell level. Not really about the discovery of new drugs. So I think anyone can find his or her interest in the technology for different purposes. It's pretty flexible.
Absolutely. No, absolutely. You're right. And in my mind, I think about it from a drug discovery perspective and the throughput. But to your point, you can very much use the pHSense and the HTRF line of products in an academic setting where you want to understand how your receptor or how mutations, for example, or expression levels of your receptor drive the pharmacology.
Definitely.
Wonderful. Any stories, any behind the scenes stories that you can share with us? Maybe one story that happened during the development of pHSense. Anything fun or not fun?
What can be fun? Honestly, it's not fun, but I can tell you when we figure out to let's move to endogenous GPCR, for me that was really a kind of cornerstone in the project. Say this is at that time. So for this, because of all what we did in the GPCR field, from the first days we said, okay, GLP-1 receptor in beta cells, this is what we have to assess.
Yeah.
But I can tell you the day the scientists from my team came in my office with the data, it was a day.
Yeah.
Because we almost immediately were able to do a dose response of exandin 4, as well as the antagonization of the agonist by the exandin 939, yes. And in a single experiment, for me, it was the proof that, yeah, we are a step ahead in what we can achieve without any imaging, without any complex experiments. And for me, this is the day I said, okay, for GPCR now, we have something almost no one else is able to do in an easy way. And that was really the game changer in my wish to push quickly the product toward the commercialization.
Sounds like a very good aha moment.
Yeah. I can tell you, yes.
Wonderful, because actually my next question is, what are the top 3 aha moments, actually now 2, that shaped the development of pHSense?
First one is succeeding because the chemistry behind the scene is not that easy. And since the beginning, we were very concerned about can we design something with the proper solubility in buffer while keeping the photophysical property. So the first key milestone was really, yes, we can do it. And I would say at a decent industrial level. So that was really the first step. And the second one was the one I was mentioning around moving to endogenous level, because I was convinced that we will succeed as soon as we get the probe. For me, overexpressed system say, okay, we should do it pretty easily.
Yeah.
It was the case. Of course, a bit of optimization and time, but really the question was, are we sensitive enough to go to the endogenous system? And when we said, yes, we are, that was the second step of, let's say, haha moment.
Wonderful. I think it's wonderful. I think when it comes to endogenous systems, it's always, and if I remember correctly, some of Jean-Philippe's work as well is around, endogenous systems and how in a natural setting, how does a GPCR actually function? So having a tool like this, and obviously having the antibody that recognizes the receptor, the endogenous receptor, without having any, agonistic or antagonistic function, is key. And being able to look at it, I can very nicely see a project where you start with your cell line, with your, HA tagged or flag tagged, and then at the end, when you're getting closer, that last figure in that paper is, on an endogenously expressed receptor in a tissue that is physiologically relevant. I think it's a beauty, all with one product.
Yeah.
Wonderful. Last question. What is your advice to junior scientists who want to work in product development just like you do?
Have fun first. That's, for me, mandatory. If you don't like, as I used to say, not everyone is able to enjoy the fact that you can try and fail. It's you need to be driven by, of course, the right theory, the right expertise, but time to time during months, you are not succeeding a lot. So still have fun in that kind of situation. And able to think a bit out of the box time to time and let the serendipity occurs. That's also a lesson from my career, is that you can frame, you can draw any strategy and anything time to time. You need to catch from what you are doing what is out of the box but seems to be right. Of course, then you need to confirm it. Then science is back, I would say, on the top of serendipity. But yes, yeah. It's a matter of mindset. Be rigorous, but not too much. That's the right balance you need to find. It's not that easy, I would say, at the end of the day. But if you are only in the rational all times, you will miss things, obviously. And try time to time something which is a bit out of the box is always good. And what is the most important, and to pick the right information, at least to check that it's real information. Time to time, it's an error or whatever. You cannot never reproduce anything that does not have any value. But I can tell you, we did time to time experiments with unexpected results. And because of the unexpected results, we move the step ahead. And you need to really to be aware that it can happen.
No, I agree. And I think it comes with experience. It comes with, you develop that sense. That's spider sense almost, where you're like, this is an unexpected result, but it got me thinking about something new.
Definitely.
Wonderful.
And I think it's underrated by many people.
I think so, but maybe because we don't talk about it enough, or maybe it's something that people, especially someone who's accustomed to it, they do it, but they don't label this sense.
Yeah.
You're right. I'm assuming you teach this to everyone on your team.
I'm trying. That's not that, again, it's a matter of balance. Depending the stages of the project, there is a lot of, I would say, it's a matter of circumstances. But in early stages, yes.
Yeah.
Let's have the right frame, but in it, let's try. If you narrow down everything to a single kind of theory, I think it's detrimental to the outcome. You need to be broad at the beginning and narrow down as much as you can. A product, you cannot afford to go everywhere. But before the product, as we discussed, there is many months or years during which you have to make the right decision in terms of what to try, what not to try, be aware of any kind of data and proper analyzing it. That's key.
I think it's wonderful. And you just mentioned this, this funnel, as you were telling the story of how pHSense came to be. That's exactly the image I had in mind. It's this funnel of, we have these probes, we modified them, and what can we do with them in order to serve a community? Wonderful. Eric, thank you so much for your time. I really enjoyed talking to you.
You're welcome. That was a pleasure.
Thank you so much. Don't go anywhere.
Okay.
Huge thanks to Dr. Eric Trinquet for joining us. His story, from the early days of HTRF to the breakthrough with pHSense, is a reminder that real innovation takes time and collaboration. That moment when data showed internalization at endogenous levels, that's what we work for. Not just clean curves, but clarity. If this episode sparked something in you, share it with a colleague. Join the Dr. GPCR ecosystem. And don't forget to explore pHSense and Revvity's GPCR portfolio by clicking on the link in the description. And for deeper insights on GPCR pharmacology, check out Terry's Corner. Don't forget, Dr. GPCR Premium members get exclusive discounts when they join Terry's Corner. And a special thank you again to our partners, Revvity and Celtaris Research, for advancing what's possible in GPCR drug discovery. This episode was made possible by the Dr. GPCR team and by listeners like you. Links to everything we've talked about are going to be in the notes of the show, because receptors don't work alone, and neither should you. Until next time, stay safe.